So we are talking about the anaphase stage, during which the sister chromatids separate from each other through assistance of the spindle fibers which pull them apart. And then we see them being pulled towards the pores, the spindle, the opposite spindle pores. So yeah, they are pulled towards the pores like that. And then we have the stage of telophase. So during telophase, the two sets of chromosomes are the spindle pore. They are at the spindle pore, and they begin reverting to their decondensed state. They begin to revert to their decondensed state. So the nuclear envelope also begins to reappear around each set of the chromosome, each set of the daughter chromosome. So remember the nuclear envelope, which had disappeared, the nuclear membrane which had disappeared in the prophase stage, the nuclear envelope is going to begin to reappear around each of the daughter chromosomes during the process of the telophase. So there will be a ring, that barrel-like contractile ring, which produces this cleavage furrow. So this which contains actin and myosin filaments, the actin and myosin filaments. So developing at the periphery of the cytoplasm, or develops in the peripheral cytoplasm at the equator of the cell. So this is a cleavage furrow. Cleavage furrow. So we have the telophase state there being depicted. So in the telophase phase, we see that the chromosomes will be at the opposite pores. And remember, I was saying that they begin to revert to their decondensed state. So meaning that they are going to start uncoiling. The chromosomes will start uncoiling, and then they're going to become thin. They'll start becoming thin. Remember, when they were in the condensed state, they started thickening. There was a thickening of the chromosome during the process of prophase. There was coiling and thickening. During the process of telophase, there is uncoiling and the becoming of thin, or the thinning of the chromosome. And the nuclear membrane is going to reappear. And then we see that each set of the chromosomes will actually be enclosed in a separate nucleus. It's going to be separated enclosed in a separate nucleus. So that's the two nuclei that form. So the two nuclei that form. And then we have we see that as it progresses until the cytoplasm and the organelles are divided into two daughter cells, and each with one nucleus. So remember, the two daughter cells are separated through a cleavage furrow. In animals, it's called a cleavage furrow. In plants, we call it a cell plate. In plants, it's known as a cell plate. So at the end of mitosis, what we see is that two daughter cells are produced, and each of them has the same number of chromosomes as the mother cell. So at the end of mitosis, the two daughter cells that are produced will each have the same number of chromosomes as the mother cell. So this is what we call cytokinesis. That's what we call cytokinesis. The progress until the cytoplasm and its organelles dividing into two daughter cells, each with one nucleus. That's cytokinesis. So today we've looked at the cell cycle. We've looked at mitosis, right? We looked at the cell cycle. We looked at mitosis. And this is simply depicting what we've actually looked at, the cell cycle and mitosis as well. So I hope you've learned the process of cell division and the process of mitosis, so that next time we meet, we're going to look at meiosis. It's a type of cell division which results in the production of special cells that we call the gametes, and these are involved during sexual reproduction. So thank you all for your attendance.